Hello and welcome to the Certara webinar on What's New for SEND 2023, part of the ongoing series, Getting the Most Out of Your SEND Data. My name is Peggy Zorn and I am Senior Director of Product Management at Certara, focusing primarily on the SEND Explorer dataset review and visualization tool. Just a little more information on me. I have a strong background in non-clinical data management, warehousing, and the SEND standard, including serving as the lead of the CDISC SEND controlled terminology team for 7 years. I continue to be involved in the CDISC SEND team, as well as the FUSE non-clinical working group. Prior to joining Certara, I worked for 15 years at Pfizer, with 6 of those focused on non-clinical data management and warehousing. I've also worked for INDS for several years, which is the company that originally developed the SEND Explorer dataset reviewer and visualization tool. In today's session, we'll be covering updates to the SEND standard for this year, 2023, and what those changes mean for staff that produce SEND datasets, and also the scientists that use SEND formatted data to analyze and interpret study results. These changes include a new version of SEND, version 3.1.1, the introduction of the requirements for SEND for submissions to CBER, expansion of the requirements for the SENDIG-AR version 1.0 for animal rule, the introduction of the requirements for SENDIG version 1.1 for developmental and reproductive toxicology or DART studies, and the requirement for the new version of the Define.xml standard version 2.1. We'll end the session by taking a look at what lies ahead for next year and beyond for SEND, as major additional changes are on the horizon with SEND version 4.0, the next major release for SEND. SEND is the standard for exchange of non-clinical data. It is a CDISC standard based on the SDTM model and covers the study design and individual animal data for in-scope non-clinical studies. Like SDTM datasets, SEND datasets are produced in SAS transport or XBT format. The current versions of the SEND standard in use at FDA now are SEND version 3.1, SEND version 3.1.1, SENDIG for DART version 1.1, and SENDIG-AR version 1.0. SEND datasets are required for single dose, repeat dose, carcinogenicity, cardiovascular and respiratory safety pharmacology, and embryo-fetal development studies with start dates after those published in the FDA Data Standards Catalog. SEND is required by both CDER and CBER. The requirement for CBER took effect on March of this year, and SEND is now required for in-scope studies and data types for studies starting after March 15, 2023. The best resource for determining the requirement for SEND datasets and associated data standards for FDA is the FDA Data Standards Catalog. This document is an Excel spreadsheet that lists all the submission data standards relevant for FDA. Each data standard version is in a row in the spreadsheet, and columns include details about the data standards such as exchange or file format, standards development organization responsible for the standard, such as CDISC, ICH, etc., implementation guide name and version, the FDA center or centers the standard applies to, and key dates associated with when support and requirements for the standard begin and end, and any details associated with the requirement dates, such as specific dates associated with requirements for NDA versus IND submissions. This file is updated frequently to add new standards, associated dates, and details to help industry better understand the requirements, so always make sure that you're using the current version. This is a screenshot of the FDA Data Standards Catalog published on April 19th of this year, filtered to include non-clinical study datasets and Define.xml standards only. You can see here the columns present, including the date columns. Keep in mind there is a difference between the date that support for a standard begins and ends and the date the requirement for a standard begins and ends. FDA support for a standard means that if a sponsor includes data following this standard in a submission, FDA can use it, but doing so is not mandatory. FDA requirement for a standard means that use of the standard is mandatory for the conditions set forth in the catalog, and the link to the statutory, regulatory, or guidance authority sources associated with the requirement is included for reference. Note, for many of the standard versions, the requirement dates are different for IND versus NDA submissions. As of March 15, 2023, there were several standard version requirements enacted that are related to SEND and non-clinical data and submissions. SEND 3.1.1, the latest version of the SEND standard, is required for studies with start dates after March 15, 2023. In addition, SEND 3.1 or SEND 3.1.1 datasets are required for submissions to CBER for studies starting after March 15, 2023. Previously, only CDER submissions required SEND datasets. The SEND Animal Rule Standard version 1.0 also became required for IND submissions for studies starting after March 15, 2023. The requirement for this standard for studies included in NDA submissions started last year in March. Another big change this year is the beginning of the requirement for SEND for developmental and reproductive toxicology studies, or DART. The DART version 1.1 standard for embryo-fetal development studies is now in effect for studies starting after March 15, 2023 that are included in NDA submissions. The requirement for IND submissions for DART version 1.1 will take effect next March. And finally, the requirement for version 2.1 of the Define.xml standard also took effect on March 15th of this year. Define.xml files are required as part of all SEND dataset submission packages. Of note, both SEND 3.1.1 and Define.xml 2.1 are now required for studies starting after March 15, 2023, but because support and requirement end dates have not been published yet for the prior versions of the SEND or Define standards in the FDA Data Standard Catalog, either SEND version 3.1 or SEND version 3.1.1 or Define 2.0 or Define 2.1 are acceptable, although the latest required versions of the standards are preferred. As you can see, a lot is changing for SEND and non-clinical submissions in 2023. We'll be going over each of these changes in more detail in this webinar. SENDIG version 3.1.1 was originally released in March of 2021 and primarily covers changes to the pharmacokinetic concentration and pharmacokinetic parameter domains, or the PC and PP domains, to ensure that creation of time concentration curves could be done by FDA using the data from these two domains. To support this, there are changes to variable permissibility, assumptions, and examples, including cross-domain examples, to make it clear what data is expected to be populated in order for the data to be used as intended. Most notably, the PC ELTM and PC TPT-REF variables were changed to expected rather than permissible, and the PC DTC variable was changed from expected to permissible. This is consistent with the FDA Technical Conformance Guide statements related to the PC domain and variables used to support creation of the time series graphs and automatic calculation of the pharmacokinetic parameters from sets of related plasma concentrations. In addition, the unscheduled flag variable was added to the PC domain to support flagging of unscheduled results. As mentioned previously, at this time, both SEND 3.1 and SEND 3.1.1 are listed as required in the FDA Data Standards Catalog, with no support or requirement end dates for SEND 3.1, so both versions are acceptable for submission. For the most part, changes outlined in SEND 3.1.1 can be implemented in SEND 3.1, so it is recommended that if you are continuing to use SEND 3.1, you adopt the changes recommended for the PC and PP domains in the SEND 3.1.1 version and the FDA Technical Conformance Guide, so even data submitted in SEND 3.1 format can be used to create the time series graphs and calculate the TK parameters. Up until this year, the SEND data requirements only applied to submissions to CDER. As of March 15, 2023, the SEND standard also applies to submissions to CBER as well. The study and data types included for CBER are the same as for CDER, with the exception of the requirement for SENDIG DART version 1.1 for embryo-fetal development studies, which are not required for CBER. In-scope study types for CBER include single, repeat dose, carcinogenicity, and cardiovascular and respiratory safety pharmacology studies. Data types required are those modeled in Codex version 1.0 for SEND 3.1. FDA has included information in the Technical Conformance Guide specifically related to handling of immune response data for CBER submissions in SEND 3.1 or SEND 3.1.1 format, as it is not modeled in these versions of the SENDIGs, but will be included in the next version as the immunogenicity specimen assessments domain is planned to be included in SEND version 4.0. In the meantime, CBER would prefer that immune response data be included in either the LB domain or in the IS domain as a custom domain for SEND 3.1 or SEND 3.1.1. Custom domains are supported in SEND starting with version 3.1. Alternatively, per the FDA Technical Conformance Guide, it is also acceptable for immune response data to only be submitted as part of the tables included in the study report. When immune response data is included in SEND datasets, this allows the use of SEND analysis and visualization tools for these endpoints. This is an example of immune response data included in both the IS and LB domains in SEND 3.1 and graphed together using the SEND-Explorer data analysis and visualization tool. In this screenshot, both endpoints are graphed over time with separate plots for males versus females. Dosing days are also depicted with black triangles along the x-axis. This is a screenshot showing anti-drug antibody data formatted in SEND in a custom IS domain using modeling from the FUSE ADA white paper, which provides recommendations on modeling of anti-drug antibody data for both SEND 3.0 and SEND 3.1. This is an excellent resource to use when reviewing options for providing immune response data in SEND format. This visualization contains summary data by sponsor group code, sex, and dose, pivoted by time point with an interactive thumbnail visualization for each time point that shows individual data plotted as well as the group mean value as the black dash. Subject data across each graph can be highlighted and the thumbnails can be enlarged for easier viewing and copying into presentations or other documents. SEND Implementation Guide for Animal Rule version 1.0 became a requirement last year for studies starting after March 15, 2022 that are included in NDA submissions. As of March 15, 2023, the requirement is now extended to IND submissions as well. Studies submitted under the Animal Rule are different from general toxicology studies in that the Animal Rule applies to using adequate and well-controlled studies in animal models rather than in humans for situations where conducting clinical trials in humans is not ethical. These conditions are life-threatening and related to exposure to lethal or permanently disabling toxic chemical, biological, radiological, or other nuclear substances. While typical toxicology studies submitted as part of NDA or IND applications focus on testing toxic effects of treatments, Animal Rule studies focus on natural history of diseases and testing of animal models as well as efficacy of treatments in animal models. The SEND-IG for Animal Rule version 1.0 introduces several new domains and variables into the SEND model, as well as updates for SEND-controlled terminology. Also added in March of 2023 is the requirement for SEND datasets for embryo-fetal development studies started after March 15, 2023, submitted in NDA applications to CDER. The requirement for INDs will take effect next March. Embryo-fetal development studies are modeled in the SEND Implementation Guide for Developmental and Reproductive Toxicology, or DART, version 1.1. This IG contains new domains for pregnancy and fetal findings, new timing variables to track results relative to gestation start, and significant additions to SEND-controlled terminology requirements. The FDA technical rejection criteria is not currently implemented for ECTD Module 4.2.3.5.2, which is where embryo-fetal development studies are located. So simplified TS-XBT domains do not need to be submitted to indicate SEND datasets are not required for the study. If you do submit a simplified ts.xbt domain in this ECTD module, FDA has confirmed that it will not interfere or prevent electronic receipt or validation of the submission. FDA has also provided clarification on requirements for SEND-IG DART version 1.1 in the March 2023 Technical Conformance Guide. The clarifications are very helpful in determining specific SEND requirements for common development scenarios. Of note, if a preliminary embryo-fetal development study is being used to assess clinical safety in lieu of a definitive study, then SEND is required. However, studies that are conducted in pregnant or non-pregnant animals only to establish dose or dose schedules for an EFD study, for example, dose range finding, tolerability, or pilot studies, are not designed to inform clinical safety and therefore do not require SEND. In addition, for combination studies such as fertility and early embryonic development, or FEED, and EFD studies, or pre- and postnatal and EFD studies, only the EFD-related endpoints that are modeled in SEND-IG DART version 1.1 require SEND datasets. Dedicated juvenile animal studies that typically include multiple phases are not currently modeled in SEND, so also would not require SEND datasets. Now that we have EFD study data in SEND, this gives us the opportunity to use SEND analysis and visualization tools to look at the data. The screenshot here is a summary visualization from SEND-Explorer for the pregnancy results domain from an embryo-fetal development study modeled in SEND-IG DART version 1.1. From the dataset, mean values for each test can be calculated and a thumbnail visualization produced to show the trend by dose group for each test. This is another screenshot of a summary visualization in SEND-Explorer for fetal findings modeled using SEND-IG DART version 1.1. Again, summary statistics are calculated for each dose group for abnormal findings category, and a thumbnail visualization generated to show the trend by dose group. Define.xml version 2.1 is now an FDA requirement for studies starting after March 15, 2023. This new version of Define.xml is similar to version 2.0 with a few notable changes. In Define version 2.1, more than one data standard can be referenced using the new Def Standards element. This means for SEND studies where you may be using SEND version 3.1 for most domains, but SDTM for a custom domain, both standards could be referenced. Another new concept introduced in Define.xml version 2.1 is subclass, which allows programmers to provide a more descriptive classification of their datasets. As of today, the only defined use case for this concept exists for ATOM submissions. Another change that came with Define.xml 2.1 has to do with identifying empty datasets and variables with no values. This is flagged with a new attribute called def has no data on both the item group def element for datasets and the item ref element for variables. This optional attribute is only needed when the condition is met, for example, the object contains no data, and will therefore have a value of yes. In addition, whenever a dataset or variable is flagged as being empty, a comment becomes conditionally required to explain why no data is present. When this flag is set, the Define.xml style sheet will show no data next to the dataset or variable to indicate that it is empty. Origins were also enhanced to include both type and source, although for SEND datasets only origin type is used. A full list of changes in Define 2.1 is included in the Define 2.1 specification, available from the CDISC website. As you move to use of Define 2.1 for your submissions, ensure that the validator tool you're using has support for this standard. Pinnacle Community and Enterprise versions do include support for Define version 2.1. It is acceptable to submit Define version 2.0 or 2.1 in submissions until support and requirement end dates are published in the FDA Data Standards Catalog for Define 2.0, although the latest required versions of the standards are preferred by FDA. This is a screenshot of what the Define 2.0 file looks like using the updated style sheet for a SEND dataset package where both the SENDIG and SEND-CT versions have been specified. Notice the table listing the two standards at the top of the page, directly below the study number and title. In addition, the standard used for each dataset is indicated in the dataset table. This screenshot shows a Define file representation of the no data tagging for variables included in the dataset that don't contain any data. The unscheduled flag for the DS domain is not populated as there were no unscheduled deaths in this study and includes the no data comment below the variable name. For 2024 and beyond, there are already two changes on the horizon related to developmental and reproductive toxicology. First, SENDIG DART version 1.1 will be required for studies starting after March 15, 2024 that are included in IND submissions. In addition, SENDIG DART version 1.2 is scheduled to be released to the public in mid-2023 and will include modeling and examples for general toxicology studies conducted on juvenile animals. This will include the addition of the developmental milestones or DP domain, as well as new timing variables relative to postnatal start date. It is not yet known when this updated version of the DART IG will be supported or required by FDA, but when that does happen, it will be posted in the FDA Data Standards Catalog. When we look ahead for SEND, the biggest change coming is related to finalization and implementation of the next major version of SEND, version 4.0. This version is still in development with CDISC, but is currently projected to reach the public review stage in the third quarter of 2024. This version of SEND has significant changes, including the addition of 8 new domains covering in vivo genetic toxicology, cell phenotyping, immunogenicity, ophthalmology examinations, pharmacokinetic inputs, nervous system test results, skin test results, and scoring scales. Additional changes planned to the existing standard include update of the microscopic findings domain to include targeted staining, sexual maturity and reproductive cycle results, new variables for result modifiers, category and subcategory, and time to detection, as well as depreciation of the tumor findings domain. At this point, CDISC final publication date for SEND version 4.0 is planned for April 2025, although this could certainly change based on feedback from the public review comments and other factors. For more information regarding SEND version 4.0 or to get involved in development and finalization of this standard, visit the CDISC SEND website listed in the resources slide of this presentation. This is a list of helpful FDA, CDISC, and Fuse SEND dataset and Define.xml resources to help with all the changes to SEND standards that have occurred so far in 2023. That's it for the topics for this presentation. Now let's move on and answer questions you all may have. First question. For studies submitted to CBER, are ADA and other immune response data required to be included? Per the FDA Technical Conformance Guide, ideally CBER would like to see this data submitted in either the LB or IS domains, even though it is not currently modeled in SEND 3.1 or SEND 3.1.1. If the IS domain is used, it will be a custom domain for SEND 3.1 or SEND 3.1.1, which is supported by those standards. Per the FDA TCG, it is also acceptable to just include this data as part of the study report tables and not in the data sets. Second question. Do you know when FDA plans to make SEND for other reproductive toxicology study types required? Well, SEND DART IG 1.2 is the next thing that has the potential for introduction of new requirements for reproductive toxicology studies to SEND, and it only includes support for juvenile animals and general toxicology studies. Other study types are being looked at by the CDISC SEND repro team, but are not yet scheduled for proof of concept or public review activities, so they're further out on the horizon in terms of expected publication. Next question. When is SEND 4.0 expected to be required by FDA? Well, at this point, SEND 4.0 is planned to be published by CDISC as final in April of 2025. After this, FDA would still need to update the data standards catalog with support and requirement start dates, so no real dates can be projected yet on when FDA will require SEND version 4.0. Thank you for your questions. If you submitted a question that we didn't cover today, we'll be in touch with you with a response in the next few days. This webinar is part of a larger series on getting the most out of your SEND data. Future topics planned include single study visualizations in SEND for monitoring ongoing studies, use of multi-study visualizations for comparing studies in a drug development program, leveraging historical control, and using SEND datasets for interactive scientific review. Please stay tuned for dates for these upcoming webinars. They will be posted on the Certara website and social media. If there are any other topics that you would like to see covered in the series, please let us know. Thank you for your attendance at this webinar. My contact information is listed here. Please feel free to reach out to me if you have any further questions or want to learn more about the SEND standard and tools for visualizing, analyzing, and validating SEND datasets.